HD is caused by a mutation in the Huntington gene, where a sequence of DNA letters CAG are repeated more than 36 times. This extra repetition creates faulty RNA, which holds the instructions for making proteins, and so a damaged Huntington protein is produced. DNA can be thought of as a long recipe book holding the instructions to make many dishes, or proteins. However, not all of the pages are needed to make one protein. When a cell wants to make a protein, it will first read the DNA recipe and copy all the pages into an RNA message. This RNA includes both important instructions and extra pieces of information. Splicing is like an editing process which cuts out the extra RNA pieces of information and stitches together only the necessary instructions required to make one protein. The final edited RNA is then used to make a proper protein that works in the body. Researchers from the University of California have found new clues as to how the editing process can go wrong in HD, leading to the malfunction of protein-making machinery in brain cells. It was previously shown that in HD, extra RNA pieces are somehow included in the final recipe for making proteins in cells. A recent study has now shown that the protein coded by the Huntington gene may play a key role in this confusion. This faulty splicing could lead to either a faulty protein being made, or the protein may not be produced at all. These outcomes are bad for the cells. The HTT protein is known to bind to other RNA molecules and to other proteins. If splicing is disrupted in HD, and if the HTT protein interacts with RNA and other proteins involved in splicing, could mutant HTT protein be interfering with the RNA editing process in cells? To answer this question, Dr. Leslie Thompson and her research group used mouse models of HD and post-mortem human brains to see if errors in splicing were happening. They found RNA molecules which incorrectly contained both important instructions and extra pieces. Interestingly, these errors were more common in medium spiny neurons, which are the specific population of cells in the brain that are particularly vulnerable to cell death in HD. The RNA molecules that were found to be improperly spliced were messages that were crucial for neural communication and brain development. The researchers investigated what proteins interact with the HTT protein, which may be responsible for splicing errors in HD. They found that a protein called TDP43 might be the cause. TDP43 is the chief editor of splicing. This protein has been well researched, as a mutation in its genetic code causes the neurodegenerative disease amyotrophic lateral sclerosis. In the context of HD, the type of RNA messages that TDP43 most often edits largely overlap with the abnormally spliced RNA in HD. This suggests that TDP43 dysfunction might be the root cause of splicing errors in HD. To see whether HTT's interaction with TDP43 might be preventing it from splicing RNA properly, the researchers looked at the binding between HTT and TDP43 in HD mice. They also investigated the location of TDP43 in brain cells from human HD brains. In control brains, the TDP43 was located inside an area of the cell called the nucleus, which contains DNA and is where splicing takes place. However, in the human HD brain, the TDP43 was mislocated outside the nucleus. In addition to the mislocalization of TDP43 in the cells, the researchers also found that in the HD brain, special chemical markings on RNA, called M6A, were missing. Without these special M6A marks, TDP43 is unable to identify the extra pieces of RNA message that need editing out, likely contributing to splicing dysfunction. The researchers hypothesized that in HD, the HTT protein is abnormally interacting with TDP43, keeping it out of the nucleus and not letting it perform splicing normally. This abnormal splicing can lead to faulty protein production and eventually sick brain cells that cannot communicate properly. These interesting findings may help guide the development of future HD therapeutics. TDP43 is already extensively studied in ALS, and hundreds of TDP43 targeting therapies are currently in development, which may provide promising starting points to study TDP43 in HD further. Importantly, this study would not be possible without the dedication and commitment of HD patients and families who have donated their brains for research.